Hello and welcome to this teleconference session. Today we are back with a very important topic on carbohydrate metabolism, which is part of the nutritional biochemistry course of the master's program in dietetics and food service management. So far we have talked about carbohydrate as a macronutrient which provides energy. We know that 1 gram of carbohydrate provides 4 kilocalories, and the major function of carbohydrate is the energy giving function. Now, how is this possible? What is the metabolic fate of carbohydrate? What happens to carbohydrate in the body? How it is broken down to release energy? These are, or rather this is an important aspect. That is what we are going to discuss in this teleconference session today. Well, I'll warn our learners, it's going to be a bit technical, but certainly we have simplified this entire metabolic process for carbohydrate utilization, digestion in the body. And that is what our learners will learn today in this program. And helping us or taking us through this entire carbohydrate metabolic process is an expert with us today, Dr. Geeta Trilok Kumar. She will take us through the entire reactions and the cycles which are involved in the carbohydrate metabolism. Before I invite Geeta to start with the presentation, let me give you an overview of what is going to come in this program. First, we've talked about carbohydrates as a major macronutrient which provides energy. In fact, 60 to 70% of our energy needs are met by carbohydrates. How? What happens? Any carbohydrate we eat, it is converted into the monosaccharide units. We know that glucose is the major monosaccharide. This, when it is broken down to produce energy, the process is called glycolysis. So that is the first thing which Geeta will talk about in this program. In addition to this, when our needs for energy, or when our body does not need the glucose or the energy, excess is stored in the body in the form of glycogen. Now this process of converting glucose into glycogen is glycogenesis. This is the second aspect which will be covered in this program. And if the body requires glucose back from glycogen, this is glycogenolysis. This is the third metabolic fate, or the third aspect covered in this program on carbohydrate metabolism. As you go through, you will see that many other reactions are also covered. One such important cycle is the citric acid cycle. This will be covered as well. So let us begin this session by first asking Geeta to introduce carbohydrate metabolism and start with glycolysis. Geeta.
Thank you, Diksha. When you eat food, your main component of food is starch, and starch finally breaks down to glucose. That is why the metabolism of glucose is very important in our body. As Diksha already told you that glucose metabolism begins with carbohydrate metabolism called glycolysis. The first cycle that involves glucose breakdown is glycolysis. So let us have a look at glycolysis. Now there are a few things about glycolysis I'd like to tell you, but as already Diksha has already introduced, there are many other pathways. First, we begin with glycolysis, which is the prime pathway where glucose enters and is distributed to various cycles. Now there are a few things I'd like to talk about glycolysis. Remember, it's the main pathway for glucose utilization. There is no other pathway where glucose can enter unless it goes through glycolysis. So it is the major pathway of glucose utilization as well as energy production. Energy is produced in the form of ATP. I hope you all understand what is the use of ATP. ATP is used for every process, chemical process in your body. So glycolysis begins with conversion of glucose to glucose 6-phosphate. Let's have a look at the table. If you look at this pathway, you can see that glucose is getting converted to glucose 6-phosphate. I need a pointer.
Here's the pointer.
Yeah.
Here. You can start from here.
So when glucose gets converted to glucose 6-phosphate, a molecule of ATP is used. Remember, whenever you're using kinases in any chemical reaction in the body, it usually involves ATP, GTP or any other phosphate. So the enzyme, since there is an ATP conversion here, the enzymes involved are kinases. There are two types of kinases that are utilized in glycolysis. One is hexokinase, the second is glucokinase. Now you will wonder, what is the difference between these two enzymes? Hexokinase is present in all cells of your body, in the cytosol, whereas glucokinase is only present in liver and alpha cells of islets of your pancreas. No other cell in the body has glucokinase. Glucokinase is an isoenzyme of hexokinase. Now the main function of glucokinase is only to remove glucose from the blood after a meal. That means when you are in a postprandial state, a few hours after you have digested your meal, you have a lot of blood sugar. That means there is a lot of sugar in your body because all your carbohydrates have been digested and absorbed. That is when glucokinase is essential, so that it picks up glucose from the blood and takes it inside the liver or the pancreas, where it can either be glycolized or it can be stored as glycogen. We will be talking of glycogen biosynthesis very soon, and then we'll come back to this. Now going back to the cycle, so glucose 6-phosphate is formed. Now this reaction is irreversible. Under physiological conditions, this reaction is irreversible, and it is one of the regulatory reactions of the metabolic pathway. So it's one of the main regulatory reactions. Now let us come back to hexokinase. Hexokinase is present in all cells. Now the function of hexokinase is to ensure that the glucose that enters the cell gets phosphorylated, that means converted to glucose 6-phosphate, so that it does not escape out of the cell. Once the glucose is phosphorylated, it ensures that glucose remains in the cell, so that whenever there is a requirement for energy, the cell has its own supply of glucose. If it did not phosphorylate glucose, glucose could escape out back into the blood. So this is very important. Now, if we go back to the chart or the pathway, you will see that glucose 6-phosphate is isomerized to fructose 6-phosphate. Glucose and fructose are isomers of each other, you know that. They have the same molecular weight, they have the same number of carbons, hydrogens and oxygens, but different looking formula. So they are isomers of each other. Now the enzyme that is involved is an isomerase, because it is converting one isomer to the other. It's converting glucose to fructose. This step is reversible. It can be easily reversed in both directions. Now the next important step is the step of conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. This is again an irreversible step, and being irreversible, it is another regulatory step of glycolysis. In fact, it is one of the most important regulatory steps of glycolysis. Because why do you think this is the most important step of glycolysis? Because at this stage, the glucose molecule is committed to enter glycolysis and nothing else. Until glucose 6-phosphate, it could have entered glycogen metabolism, it could have entered pentose phosphate pathway or glycolysis. Once it forms fructose 6-phosphate, there is no other way that this molecule can move. It has to go through glycolysis and produce the last molecule of glycolysis, which is pyruvate. Now going back to the pathway, let us have a look at what happens once fructose 6-phosphate is formed. Fructose 6-phosphate, if you look at the structure, I also told you in the beginning that when there is a kinase, there is an involvement of ATP. Similarly, phosphofructokinase involves the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. So it also requires another molecule of ATP. So now you have already spent one molecule of ATP here and one molecule of ATP here, which means you have already utilized two molecules of ATP. Instead of, you must be wondering, instead of producing ATP, we are actually utilizing ATP. But once it comes further down, you will see that the returns are coming in. So all that you have spent is being returned back to you and much more. Now, fructose 1,6-bisphosphate is a 6-carbon molecule. It has two phosphates, one at carbon number 1 and one at carbon number 6. Right? This molecule splits in the center, right here. Okay? It splits in the center to give you two halves of 3 molecules. So it forms 2 isomers again, one glyceraldehyde 3-phosphate, one dihydroxyacetone phosphate and another one glyceraldehyde 3-phosphate. If you see this molecule is split halfway. The first phosphate comes into the dihydroxyacetone phosphate, the last phosphate comes into glyceraldehyde 3-phosphate. So the first half of the fructose molecule is converted to dihydroxyacetone phosphate. If you look at these two structures, they are again isomers of each other. You already know that glyceraldehyde 3-phosphate and 1,3-bisphosphoglycerate are similar looking, but glyceraldehyde 3-phosphate and dihydroxyacetone phosphate have actually the same number of hydrogens, oxygens and carbons. So again, we have two sets of isomers. I mentioned before that whenever one isomer is getting converted to another isomer, an enzyme called isomerase is participating in the reaction. It is responsible for interconversions of isomers. So if you look at this pathway, the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are being interconverted by an isomerase, a triose phosphate isomerase. Okay? Remember that all molecules of dihydroxyacetone phosphate eventually get converted to glyceraldehyde 3-phosphate. So how many molecules of glyceraldehyde phosphate do we have? 2 molecules. For every molecule of glucose, you end up getting 2 molecules of glyceraldehyde 3-phosphate because you're already forming one glyceraldehyde 3-phosphate and the other one is from dihydroxyacetone phosphate. So we proceed further to look at what happens to glyceraldehyde 3-phosphate. Henceforth, we have 2 molecules of glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate moves to form 1,3-bisphosphoglycerate. Now, if you look at this reaction, it's an oxidation reaction. Again, this is not a reversible reaction. And NAD is getting converted to NADH plus H. Now, let me remind you, each time NAD gets converted to NADH plus H, this NADH plus H enters the electron transport chain. We will be doing electron transport chain later on, so you will understand what the whole process is about. But for the time being, remember that NADH plus H enters the electron transport chain, goes through a series of reaction and finally gets converted back to NAD, which comes back to the reaction. If the electron transport chain is not functional, this will not happen. In this whole process, NADH plus H gets reoxidized back to NAD, and the process releases 3 ATPs. Each molecule of NADH plus H, when it enters the electron transport chain, produces 3 ATPs. Now, the new product that is formed is 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate loses one of its phosphates to form 3 phosphoglycerates. If you notice on the diagram, the phosphate is removed and a COO group is retained, which means that the phosphate is lost. And where does this phosphate go? The ADP picks up this phosphate to get converted to ATP. So here you are getting 1 ATP. Okay? If you have 2 molecules of glyceraldehyde 3-phosphate, you get 2 ATPs here. Similarly, as I said, you get 3 ATPs from 1 NADH plus H. So if you have 2 NADH plus H, you get 6 ATPs. So we are already gaining ATPs. Don't you see that? Now this 3-phosphoglycerate, what happens to the 3-phosphoglycerate? The 3-phosphoglycerate starts getting converted to 2-phosphoglycerate. Now what is this process? It is only a shift of phosphate from the 3rd position to the 2nd position. And this occurs with the help of an enzyme called mutase, phosphoglycerate mutase. Now I would like you to remember another point. Whenever there is a shift of a group within a molecule, that means from position 1 to 2 or 2 to 3 or 1 to 3, anywhere within one molecule, usually the enzyme that is involved is a mutase. So here you see that the phosphate is being shifted from position 3 to position 2, and you have an enzyme called mutase. Next you have 2-phosphoglycerate forming phosphoenol pyruvate. Now if you recall from your basic carbohydrates, what do you mean by an enol? Enol is a 2-carbon structure, which is, if you look at a structure of an enol, it is 2-carbon structure with hydroxyl groups attached to it. A 2-carbon double bonded structure with a hydroxyl group. So can you see, if you look at this diagram, can you see 2 carbons with a double bond and a hydroxyl group? Here, right? And if you look here, you can see 2-carbon double bonded structure with a hydroxyl group. Now, which enzyme is responsible for conversion of phosphoenol pyruvate to enol pyruvate? The enzyme is called phosphoenol pyruvate. I'm sorry. The enzyme is called enolase. This enzyme is responsible for conversion of phosphoenol pyruvate to enol pyruvate. Now, enol pyruvate is very short-lived because a double bonded carbon is never stable. So this has to be immediately converted to ketopyruvate, and this spontaneous conversion is very important. Now, pyruvate kinase is the 3rd enzyme which is using ADP to ATP. If you see a phosphate molecule is getting lost, can you see it here? ATP is being formed. ATP is being formed from an ADP, and this phosphate is being added.
If you go back.
Yeah, I'd like to go.
Yeah.
So if you see this, a molecule called pyruvate is formed, which is the end point of a glycolytic pathway. We began with glucose, and end it with 2 molecules of pyruvate. Under aerobic conditions, all glucose gets converted to pyruvate and glycolysis ends here. However, sometimes what happens? You are exercising. Your muscle is getting tired. After a very long time you keep exercising, either you haven't exercised very much and your muscle is not capable of tremendous amount of exercise, or alternatively you are exercising more than what you actually can handle or what actually you have been doing so far. So as a result, the muscle starts getting tired. How does the muscle get tired? The oxygen supply to the muscle is not proportional to the muscle activity. So what happens is, when oxygen is less, anaerobic conditions get created in the muscle. Now when anaerobic conditions are created, what is happening? The electron transport chain is affected because the electron transport chain requires oxygen for its function. Now if electron transport chain is affected, we discussed in our previous slide that the NAD is not getting converted to NADH plus H. Now this NADH plus H has to go back to the electron transport chain to get reoxidized. If the electron transport chain is not functional, the NADH plus H does not get reoxidized back to NAD. So NADH plus H starts accumulating. Now, it's okay. It would have been okay if we had enough lots of supply of NADH plus H could have accumulated. But what happens is there is a limited supply of NAD in the cell. So the cell starts having lower and lower supply of NAD. This is when the anaerobic glycolysis mechanism takes over. Now if you look at the previous slide that we were looking at, pyruvate is getting converted to lactate. Why? Because the NADH plus H was getting accumulated. Now we need NAD back so that it can go here and catalyze this reaction. So what happens is the enzyme lactate dehydrogenase converts NADH plus H to NAD because it requires NADH plus H as its coenzyme. So in this process, what happens is pyruvate starts getting converted to lactate and NADH plus H gets converted back to NAD. Now this NAD goes back here to this reaction and glycolysis can continue. If this didn't happen, then glycolysis would have stopped, slowed down first and completely stopped when all this supply would have been exhausted. So it's a very good mechanism to ensure that some amount of glycolysis can continue so that your energy can be supplied to your body. Because the muscle requires energy, so some amount of muscle activity can continue. This lactate eventually diffuses out of the muscle, it goes to the blood, goes to the liver and gets utilized into other products. So this in a nutshell is anaerobic glycolysis. Let us have a look at the next slide.
Okay.
The pyruvate is getting converted to lactate in anaerobic glycolysis. NADH plus H is getting reconverted back to NAD and the enzyme responsible for this is lactate dehydrogenase. So this is all about both aerobic and anaerobic glycolysis. Now let us once go back to calculate the energy, because eventually you must know how many ATPs are produced in glycolysis. You must be wondering how, where the ATPs are being produced. So let us have a look at the production of ATP. Now if we want to calculate the moles of ATP in anaerobic glycolysis, it's very difficult because the amount of moles of ATP in anaerobic glycolysis is much lower than aerobic glycolysis. So first let us calculate the amount of ATP produced in aerobic glycolysis.
Perhaps, Geeta, we could understand this concept better if we actually go back to the glycolysis pathway, and if you could again, pinpoint exactly where the NADH is being formed and releasing.
Fine.
So if we go back and then I think it will help our learners understand how many ATPs in the end, what is the energetics of the glycolysis.
Okay.
Yes, Geeta. Yeah, so
now let us calculate the number of ATPs. If you go back to your glycolysis pathway,
yeah,
in the first step, glucose to glucose 6-phosphate, you are losing 1 ATP. So let us take it as minus 1 ATP, right? The next ATP is being lost here. So how many ATPs have we lost? 2 ATPs. Minus 1 here and minus 1 here. So let us keep in mind we have lost 2 ATPs. Here we are not gaining any ATPs but losing ATPs. Now let us come to the next step. Glyceraldehyde 3-phosphate dehydrogenase. Here, as I told you, NADH gets converted to NADH plus H, and this NADH plus H goes to the electron transport chain and gives 3 ATPs. I also told you that dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate. So we have 2 molecules of glyceraldehyde 3-phosphate. So how many ATPs? 3 for 1 molecule, 6 for 2 molecules. So we've already gained a substantial amount, which is 6. Minus 2 plus 6. So how many are we left with? Plus 4. Now let us go to the next step. Here again, ADP is getting converted to ATP. Now if ADP is getting converted to ATP, it is plus 1 ATP, right? 2 molecules of 1,3-bisphosphoglycerate, so you get 2 ATPs. So it's plus 6 again, right? Now in the step of pyruvate kinase, again 1 ATP is being converted to 1 ATP per molecule of pyruvate form. If there are 2 molecules of pyruvate form, again you have plus 2 ATP. So it's 8 ATP. So conversion of glucose to pyruvate results in production of 8 molecules of ATP.
Yeah.
That's simple enough?
Yeah. So yes, Geeta, you've given us a good idea about starting from glucose, how it is totally broken down to release energy, and we have given the energetics or the total amount of ATP released at the end. But I believe this is not the complete breakdown. I mean, this is only partial release of energy, and further reactions are also involved in this when the substantial amount of energy is released, so to say, in a laymanish way. So if we could move on to these reactions, I think which form a very important part of the carbohydrate metabolism, and it is called the citric acid cycle.
Disha, before we go on to the citric acid cycle, I would like to talk about conversion of pyruvate to acetyl-CoA.
Yes, so that is the first and the important step.
So
yes, we
let
will
us
start
go on
with that.
to conversion of pyruvate to acetyl-CoA.
Yeah.
Now, we were talking about formation of pyruvate. Now, what happens to this pyruvate? All this is taking place, the conversion of glucose to pyruvate is taking place in the cytosol of the cell, right? So the cytosol is the main area where glycolysis is taking place. Once pyruvate is formed, it immediately goes to the mitochondria, okay? Because the next step, as Diksha mentioned, the citric acid cycle has to take place in the mitochondria. But we have to prepare pyruvate to enter the citric acid cycle. And the preparation of pyruvate to enter citric acid cycle is a small set of reactions which are known as conversion of pyruvate to acetyl-CoA. This is also known as oxidative decarboxylation of pyruvate. So if you look at what is being written, you can see that the enzyme is a complex. the enzyme that converts pyruvate to acetyl-CoA is a complex. And this complex is known as pyruvate dehydrogenase complex. This complex is made up of 3 enzymes and 5 coenzymes. The 3 enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. And the 5 coenzymes that are involved in this whole process are TDP, which is thiamine diphosphate, coenzyme A, lipoate, FAD, you're all familiar with FAD, and NAD. So with the help of these 3 enzymes and 5 coenzymes, pyruvate gets converted to acetyl-CoA. It's a very technical process, so we will not go into details of the process because the mechanism is very technical, but you have to just remember that all these enzymes and coenzymes ensure that 1, pyruvate is converted to acetyl-CoA, and 2, all the coenzymes are regenerated. That means if NAD goes in, it has to be converted to NADH plus H. If FAD goes in, it gets converted to FADH plus H. But what happens if we start converting all these to their reduced forms? What would happen to the coenzymes? The same problem as we had in glycolysis. So this whole bunch of enzymes and coenzymes ensures that if FAD enters, gets converted to FADH2, this FADH2 is reconverted back to FAD. Similarly, if lipoate enters, it's reconverted back to its oxidized form, because in the process it is reduced and gets reconverted back to its oxidized form. The only coenzyme that does not regenerate in this whole reaction is coenzyme A, and that we will see gets regenerated in the citric acid cycle. So now you're clear that pyruvate gets converted to acetyl-CoA by the enzyme pyruvate dehydrogenase, which is a multi-enzyme or an enzyme complex and has several enzymes and coenzymes. Now let us go on to the citric acid cycle. I would like to remind you a few things about the citric acid cycle, and if you look at what I have put up, you will see that citric acid cycle is a final common aerobic pathway for oxidation. There is no anaerobic citric acid cycle, only aerobic. It takes place in the mitochondria because the pyruvate I told you had already entered the mitochondria. The acetyl-CoA had been formed in the mitochondria from pyruvate, and now it is waiting to enter the citric acid cycle. The citrate in the mitochondria is degraded and it releases several reducing equivalents. I hope you understand the term reducing equivalents, that is NADH plus H and FADH2. Okay? And these go into the electron transport chain, and at the end of the cycle you will see that citrate is regenerated. So unlike glycolysis, it's a cyclic process. It starts from citrate and goes back to citrate, okay, releasing carbon dioxide and reducing equivalents. I'll repeat. It starts with acetyl-CoA and oxaloacetate to form citrate, goes through a whole cyclic process, oxaloacetate is regenerated, and in the process carbon dioxide is lost and reducing equivalents are produced. Now if we look at the citric acid cycle, I would like to just remind you of a few things. This citric acid cycle is a common pathway for not only carbohydrate metabolism but even other metabolic pathways like lipid and amino acid metabolism. Of course, we will go to that in the next few lectures, but if you see, gluconeogenesis of carbohydrate metabolism utilizes citric acid cycle, transamination utilizes citric acid cycle, and so does deamination. Now, let us look at the citric acid cycle so you will become more clear on what the whole process is. I told you that acetyl-CoA is produced by the pyruvate dehydrogenase complex. This is acetyl-CoA. Now, acetyl-CoA combines with the molecule of oxaloacetate to produce citrate. Why is citric acid called tricarboxylic acid cycle? Can you think of it? Because it has 3 carboxyl groups. Can you see the 3 carboxyl groups? 1, 2 and 3. That is why it is also called as a tricarboxylic cycle. So a dicarboxylic oxaloacetate forms a tricarboxylic citrate after combining with acetyl-CoA. And this enzyme that enables the formation of citrate is called citrate synthase. Now, the next step, this enzyme called aconitase removes a molecule of water and forms cis-aconitate. Now, this product, again, just a few minutes before I mentioned that carbon-carbon double bond is highly unstable. And that is why this compound tries to form a stable form. The carbon-carbon double bond always looks at a stabler form, which is not a carbon-carbon double bond. So if you look at this, it's found a stabler form. And how has it formed a stabler form? Another molecule of water is added. The same enzyme first removes water and then adds water. Now if you look at the structure of citrate and this new compound called isocitrate, they are isomers of each other because water was removed from a different place by removal of H and OH. The OH was removed from here, the H was removed from here. And when water is added, the OH is added here, the H is added here. So the different positions where H and OH are added, that is why it forms a new molecule called isocitrate. Now this isocitrate is the first stable compound after citrate. This isocitrate again forms another intermediate which is called oxalosuccinate. If you look at this, it forms oxalosuccinate, and isocitrate forms oxalosuccinate by removal of hydrogen atoms. If you can see, 2 hydrogen atoms are removed from here. And what helps in removal of hydrogen atoms? NAD and a dehydrogenase. The enzyme isocitrate dehydrogenase, which utilizes NAD as its coenzyme, removes the hydrogen atoms from here. This is a very short-lived compound and it immediately gets converted to alpha-ketoglutarate. Again, the same enzyme is responsible for both the functions. So there is a dehydrogenation here and a decarboxylation here. We started with 1, 2, 3, 4, 5, 6 carbons here, and we have 1, 2, 3, 4 and 5 carbons here. Why? Because we've lost 1 carbon as carbon dioxide. Can you all see that? This compound alpha-ketoglutarate undergoes oxidative decarboxylation to form succinyl-CoA. Now, if you recall, pyruvate formed acetyl-CoA by oxidative decarboxylation. The same oxidative decarboxylation process is being used here. The only difference is that the enzymes are slightly different. The coenzymes are identical to pyruvate dehydrogenase complex. So this is also called as alpha-ketoglutarate dehydrogenase complex, which performs the same function as pyruvate dehydrogenase. Now, on oxidative decarboxylation, there is another loss of carbon dioxide. So now you've lost another carbon atom. How many carbon atoms are left? 4. 1, 2, 3 and 4. These 4 carbon atoms continue till oxaloacetate as 4 carbon atoms. But if you see, there is, after the alpha-ketoglutarate, you form succinyl-CoA, which is the oxidative decarboxylation, and succinyl-CoA undergoes a change where one of the phosphates is converted. It's actually, there is no phosphate group present here, but what it does is it uses ADP and produces a molecule of ATP for the conversion of succinyl-CoA to succinate. The coenzyme A is lost, and to remove that strong bond, a molecule of ADP is converted to ATP. So the new compound that is formed is called succinate. Without the, if you can see, the CoA is lost, and this CO becomes C double bond O, a carboxyl group is introduced. Okay. Now the next step is dehydrogenation of this compound, and dehydrogenation takes place at these two carbon atoms, where one hydrogen from each side is removed to form a compound called fumarate. Now you must be wondering why COO groups are written on the opposite sides and hydrogens are written on the opposite sides of a double bond. Because the compound that is resultant is a transform, and in a transform the groups are on the opposite side. This is a trans double bond, and in a trans double bond you will find that the hydrogens are on the opposite side. So naturally the carboxyls are also on the opposite side. Now fumarase acts on a trans isomer of fumarate to form malate. Now this malate is L-malate and not D-malate. Okay? And finally there is another dehydrogenation, and if you look at the structure, the dehydrogenation results in a formation of a oxaloacetate molecule. Two hydrogen molecules are removed and you have a C double bond O. This oxaloacetate re-enters the citric acid cycle.
So Geeta, we have about 2 minutes. Can we quickly tell the energetics of this?
Yeah.
Or will it take, I think we could.
I think
so
we
if
can.
you could go back to that slide and tell our learners that how many ATPs are actually formed at the end of the citric acid cycle.
Now you already know that NADs are in the electron transport chain. When they are reoxidized, form 3 ATPs. FADH2, when it goes to the electron transport chain, gives 2 ATP, and ADP to ATP is 1 ATP. So if you look at the cycle, you will see that here, in the first step, here, succinate thiokinase, 1 ATP is being produced here.
So +1.
Right? So +1. In isocitrate dehydrogenase, 3 ATPs are being produced, so +3. So 3 + 1, 4. FAD to FADH2, +2. And NAD to NADH plus H, +3. So if you total all this up, there are 12 ATPs. 3 from NAD, 1 from FAD, and 1 from ATP. Clear? You can see 3 NADHs plus H.
Yeah.
1, 2 here, and 3. So 1, 2, 3 NADPHs, which makes it 9 ATPs. 1 FADH2, which makes it 11 ATPs, and 1 ATP, which is 12 ATPs. It's very simple, straightforward calculation of energy.
So, Geeta, I think what we've done today is to tell our learners of how glucose, starting with glucose, we get pyruvate, and from pyruvate, how along with the acetyl-CoA, it releases carbon dioxide and results in production of 12 ATPs in citric acid, plus 8 in the glycolysis. So, this was part of the carbohydrate metabolism we have just talked about. In the second part, we will take up the other aspects we talked about, that how glucose, when excess glucose is there, how it is converted into glycogen, and then from glycogen reconverted back into glucose by the process called glycogenesis and glycogenolysis. So we'll be back in the second part with the carbohydrate metabolism. Thank you.